Well, welcome back, everybody, to our last meeting or our last presentation for the semester. So today we're going to be looking at Chapter 17, which shows us or discusses how we take the information encoded in the DNA in the form of a gene and use that to direct the synthesis of a polypeptide. As the title says, from gene to protein, or we could say from gene to polypeptide. So there are a number of different things that we're going to talk about today. We're going to address the two processes, transcription and translation. And one of the things that I want to remind you of is that transcription, while it looks a little bit like DNA replication, is not DNA replication. I've noticed over the last few years that there has been a tendency for students to equate DNA replication with transcription. They are not the same. Even though there are similarities between the two processes, they are not the same. They are distinctly different processes. So make sure that you are able to differentiate between what goes on in DNA replication, which we talked about in the previous presentation for Chapter 16, and transcription, as we will talk about it today. We'll talk about the genetic code and some aspects of the genetic code that are important to understand. And then we'll focus very specifically on each of the two processes, transcription and translation. Now, I am going to include a little bit more information in the presentation than is actually featured in the chapter, because I'm going to draw from another chapter and talk a little bit about how transcription is regulated, because the way it's presented in the textbook doesn't go far enough for you to be able to understand very easily the differences between transcription in bacteria and transcription in eukaryotes. So I want to add a little bit of extra information so that you can clearly differentiate between the two processes, because they are very different. As is termination of transcription. And I'll talk a little bit more in detail about that when we talk about transcription as well. Then we'll talk about translation, the actual assembly of polypeptides. And then finally, we'll talk about the effect of changing nucleotide sequences, the effect of mutation. So we've got a reasonable amount to get through today. And of course, along the way, there will be breaks with concept check questions. And I would encourage you again to utilize those breaks to take a step away, let your brain clear a little bit before you continue on. I may very well step away myself from the computer. I've discovered that sitting here and recording is a little bit of a chore sometimes, and I really do need to get up and walk away. You won't notice, but I would encourage you to do the same thing. If you find that you're starting to suffer from information overload, it's a really good thing to step away, let your brain clear a little bit, and then come back again. So that being said, let's get started. So we come back again to a very important time point in talking about the role of DNA, 1901. Again, we're beginning with the recognition of Mendel's work. Remember, that took place around 1901. 3 individuals independently recognized the importance of Mendel's work. And just to give you a heads up on this one, at least 2 of them recognized the importance of Mendel's work by repeating Mendel's work and claiming it as their own. Yeah, and that actually was what got them caught out, because other people said, hey, wait a minute, someone else has done that already, and recognized that it was Mendel's work. And the fact that people repeated Mendel's work and tried to pass it off as their own really forced the biological world to take another look at Mendel's work and recognize exactly how important it was. And it really is probably and I'm struggling to find the right words here, and I'm not sure why. it probably exemplifies just how important people took Mendel's work at this point in time when we recognized the very first study to look at the inheritance of the human condition was actually first published in 1902, exactly 1 year after Mendel's work was finally given credit. And this was by a British doctor named Archibald Garrod. And we'll talk a little bit about Garrod's work because he was very much a man way before his time. The insights that he had probably put him about 30 or 40 years ahead of everybody else. We do recognize the significance of what he did, but at the time, I suspect he pretty much suffered the same fate as Mendel. Very interesting, but not really that important at this point in time. So one of the things that becomes significant, once you recognize that not only is the DNA passed from one cell to the generation of cells to the next generation of cells, or from one generation of organisms to the next generation of organisms, once you start to recognize that DNA is the molecule of inheritance, then you really have to start thinking in terms of, well, what is it that the DNA is actually responsible for? If there is information encoded within the DNA, what is that information all about? That was something that Watson and Crick actually did not address in their paper, because no one at that point in time really understood the significance of what that information in the DNA molecule really stood for. It would take a while for people to actually recognize what that coded information stood for. And it would take 12 years of very difficult work to try and figure out exactly what the genetic code represents, and that the genetic code actually codes for amino acids that get assembled into polypeptides. Was that suspected? Yes, there was a suspicion that that was the case, and we'll talk about an experiment that indicates that the connection between genes and proteins or polypeptides or enzymes at various times, each were proposed as a possible product of the gene. But it wasn't until the genetic code was cracked and it was confirmed that, in fact, each of the members of the genetic code, each of the units within the genetic code actually coded for very specific amino acids, that you could confirm that the information in DNA really did code for the expression of polypeptides or the synthesis of polypeptides. You had to make that connection. And that connection was not finally confirmed until the mid-1960s, when the genetic code was fully fleshed out. We do talk about the two processes that direct the synthesis of polypeptides using language terms. And the reason why is because we use the analogy of language to talk about the information coded in a DNA molecule versus the polypeptides. We say that the cell, and by extension, of course, all organisms, speak two languages: the language of the nucleic acids, DNA and RNA, written in the form of nucleotides, and the language of amino acids, which is the language of polypeptides, written in the form of 20 common amino acids. And in terms of transcription, the term transcription simply means changing from one form of language to another form of language, which is exactly what you do when you make a messenger RNA copy of the DNA. You're writing it still in nucleotides. Both DNA and RNA are polynucleotides, so we're writing it still in nucleotides. But as you should remember, DNA and RNA are two different types of nucleic acid, because the nucleotides do have differences, which you should be aware of. And so you really are going from one version of the nucleotide language to another version of the nucleotide language. And when you change from one form of language to another form of language, like making notes based on what I'm saying here in these narrated presentations, you're doing what we refer to as transcription. You're transcribing my spoken words into written words. That's why we use the term transcription. When you change from one language to another, so from the language of nucleotides to the language of amino acids, which is what you do when you read off the information in the messenger RNA and use that to assemble polypeptides, you're going from the language of nucleotides, the messenger RNA, to the language of amino acids, the assembled polypeptide. You are changing languages. And of course, anyone who speaks more than one language realizes that the requirement for translation. And so we talk about this process of assembling polypeptides as being translation. And we talk about a flow of information. And this was first proposed in the early 1960s by Francis Crick, when he, in a short paper, outlined what he would refer to as the dogma of molecular biology, that the flow of information in a cell is from DNA to RNA to protein. And this has been the accepted flow of information in cells ever since Crick first proposed this. We do know that we do have to amend that a little bit on the basis of what we've seen, particularly with viruses like the HIV virus, which is an RNA virus, because we now know that it is possible to take an RNA molecule as a single-stranded polynucleotide and utilizing an enzyme called a reverse transcriptase, create a single-stranded DNA molecule that is complementary to that single-stranded RNA molecule. At that point, then you can then use the single-stranded DNA molecule as a template to generate a double-stranded DNA molecule. And this is in fact what we know happens when an HIV virus infects the CD4 cells of our immune system. That double-stranded DNA, which represents now a copy of the original RNA molecule that was contained within the HIV virus, then inserts itself into a chromosome in the nucleus. And that allows the HIV virus to hang out inside cells of the immune system and escape the recognition of the immune system until such times as the virus reactivates again and eventually destroys the immune system and leads to the onset of acquired immune deficiency syndrome, otherwise known as AIDS. So there is a modification of this process. We do know that we can go from RNA back to DNA, but the general flow of information is still considered to be DNA to RNA to protein. And this is essentially universal in every cell. And certainly at the cellular level, this is the flow of information. So the processes of transcription and translation still apply as first outlined, not so much by Crick, but by people who followed on as they tried to figure out the nature of the flow of information from DNA to RNA to protein. So when we start thinking in terms of this flow of information, the first person that we really talk about is Garrod. Now, Garrod first published a paper about the condition that he was looking at, known as alkaptonuria, and it's actually the subject of one of my first concept check questions. So I won't tell you very much about alkaptonuria yet, because I don't want to give away too much until we get to the concept check question. But in 1909, on the basis of everything that Garrod had observed about this condition that he was studying, alkaptonuria, which he recognized was an inherited condition because it was found in multiple generations of the families that he was studying, which clearly indicated that it was an inherited condition, and that's why he started to apply Mendel's principles to his study of this condition. And one of the things that he started to recognize was that the nature of alkaptonuria suggested that it was actually a metabolic condition, an inherited metabolic condition. So what does alkaptonuria cause? Well, it causes a change in the color of your urine. You know that the natural color of urine is usually a light to dark yellow, depending on whether it's the first pee of the morning or a pee later in the day. It's usually very dark yellow, almost orange, first thing in the morning because of the concentration of urea and uric acid in the pee. But later in the day, it gets a lighter yellow. And the reason why it has that color is because of some metabolic reactions that occur that produce the urea and the uric acid. In alkaptonuria, and this sounds a little bit scary, your pee is black. That's what it looks like. It's black in color. And Garrod realized that the problem must lie in one of the reactions that led to the presence of urea and uric acid in the urine of the individuals that he was looking at. And because he knew that this was inherited, and because people were starting to think in terms of Mendel's inheritance particles, and the term gene was starting to be used for these inheritance particles of Mendel's, and people were already starting to think in terms that they were on chromosomes. Morgan hadn't confirmed that yet, but people were fairly confident that genes were on chromosomes. It was the only way to explain Mendel's observations. And so Garrod started thinking about, well, what does that mean about a gene? What is its connection to what I'm seeing in these people who have alkaptonuria? And he realized, because he had a very strong background in the latest biochemistry, that this must be geared to an enzyme that was involved in a chemical reaction occurring in or not occurring in the individuals who suffer from alkaptonuria. And he made the connection that a gene must be expressing its action through an enzyme, that the phenotype of alkaptonuria, the black urine, was a product of a faulty gene that was producing a faulty enzyme that meant the final conversion to urea and uric acid was not taking place and the urine was black. Now, it would subsequently be shown nearly 30 years later that that was in fact a connection between genes and enzymes, that genes do direct the synthesis of enzymes. And we'll look at that experiment in a moment. But this is why Garrod was and still is considered to be so far ahead of his time, because he made this connection between genes and a genetically inherited condition and the phenotype that was associated with that condition, and indicated what he thought genes were doing, something that people were not really thinking about at that time, but would subsequently have to think about. And so when we start looking for the evidence that confirms what Garrod had stated back here in 1909, we have to skip forwards into the 1930s and the work of two individuals named Beadle and Tatum, who worked at the University of California in Berkeley. Both of these individuals had connection back to Morgan. This is another one of the reasons why Morgan, it has to be considered as the father of modern genetics in the United States, because so many people who went on to do a lot of genetics research got their start either working with Morgan directly in his lab or working with people who had worked with Morgan. And Beadle and Tatum belonged to that group of individuals. Both of them had connections back directly to Morgan's lab. Now, when they chose to do their genetics research, rather than competing with Morgan and working with fruit flies, because Morgan pretty much had the fruit fly scene wrapped up, they chose to work with a fungus called Neurospora crassum. This is a member of the same family of fungi that we refer to as molds. Now, a mold is simply a fungus that reproduces by releasing spores and growing on the surface of the materials without exchange of genetic information. But we do know that Neurospora and most molds also reproduce sexually. And one of the things that Beadle and Tatum were going to do was actually do genetics experiments, doing crosses by exchanging genetic material with the Neurospora crassum, but only after they had mutated it using X-rays. Because at this point in the mid-1930s, it was known that X-rays could create mutations. This is why it was so unfortunate in the case of Rosalind Franklin that she chose to ignore what was known about the effect of X-rays and not to take precautions while she worked with them. And as we talked about in the previous presentation, that did not serve her well because it is almost certain that her cancer was a product of her exposure to X-rays. But Beadle and Tatum were going to take advantage of the fact that X-rays would create mutations, and they were going to then examine these mutations for their ability to grow or not to grow on what is referred to as minimal medium. A minimal medium is simply one that provides the minimal chemical support that the organism requires, in this case the fungus, in order to grow. And so in the case of Neurospora, the minimal requirements are that it requires a little bit of salt and a couple of other minor nutrients. You give it that and it's off to the races. It can produce anything else that it requires. So once you start mutating it, Beadle and Tatum assumed that if they were to create the correct mutations, the fungi would no longer be able to grow on minimal media, and they would have to add other supplements in order to get the fungus to grow. And by looking at the sort of supplements that they had to provide, they could start to get a window on what metabolic processes were being affected. And the one that they really started to work with was the metabolic process that leads to the production of the amino acid arginine, something that the wild Neurospora crassum can produce all by itself, so long as it has the supplements required for the minimal media, just the salt and a few other minor nutrients, a vitamin and a couple of other things. It can make anything that it needs. So while the actual metabolic pathway to arginine was not fully developed at this point, it was well known that the molecules ornithine and citrulline were intermediates along the way to making arginine because of their structure. And what was known was that there must be some form of organic precursor molecule that the fungus was able to make, that it then converted to ornithine. So that would be a biochemical step, that would be a chemical reaction. The ornithine was then converted to citrulline, a second biochemical step. And finally, the citrulline was then converted to arginine, the third chemical step. And so what Beadle and Tatum assumed, and as it turns out, they were reasonably correct in this assumption, each one of those biochemical pathways has an enzyme associated with it. So the first chemical reaction, precursor to ornithine, they assigned to enzyme A. They're just using random labels here. They don't know what the enzyme actually is, but they're just calling it enzyme A. Enzyme B then catalyzes the conversion of ornithine to citrulline. Enzyme C then catalyzes the conversion of citrulline to arginine. Once they started looking at the mutations and that they developed using the X-rays, they recognized that all of these mutations and the mutant fungi that contained these mutations could basically be grouped into one of three different classes based on what supplements you had to provide in order to get the fungus to grow. So remember, the wild type doesn't require any additional supplements other than the basics of the minimal media. So the first mutant, what they refer to as the class 1 mutant, requires ornithine in order to be able to grow. Once it's supplied with ornithine, then it will produce citrulline and produce arginine. Of course, if you provide it with citrulline, it will still grow. If you provide it with arginine, it will still grow. But if you don't provide it with ornithine, it won't grow. And that indicated to them that the problem was in the conversion of the precursor to ornithine, whatever that precursor was. And because that chemical reaction was not occurring, they said that the problem must lie with enzyme A, because that's the enzyme that's carrying out the conversion. If the conversion doesn't happen, it's likely that enzyme A is the problem. The second class of mutants, the class 2 mutants, would not grow unless you provided them with citrulline. It didn't matter if you provided them with ornithine, they wouldn't grow. You had to provide them with either citrulline or arginine. And that indicated to them that the problem must be in the conversion of ornithine to citrulline. Once you give it citrulline, it's fine. You give it arginine, it's fine. You give it ornithine, it's not fine. So the problem lies with the conversion of ornithine to citrulline. And they assigned this fault to enzyme B. That must be the enzyme that was faulty here. The third class of mutant, the class 3 mutants, would only grow in the presence of arginine. It didn't matter if you gave it ornithine, it didn't matter if you gave it citrulline, it would only grow if you gave it arginine. And so Beadle and Tatum assigned that fault to enzyme C, the enzyme involved in converting citrulline to arginine, because that was where the problem obviously lay. It simply could not convert citrulline to arginine. Now, they did double check to make sure that, in fact, the class 2 mutants could convert precursor to ornithine, and it could. And they did check to make sure that the class 3 mutants could convert precursor to ornithine to citrulline, and it could, confirming that the problems lay with the enzymes that they suspected were the ones at fault here. Then they had to start thinking about where these faults came from. Obviously, the faults lay with the enzymes, but those faults only occurred because they'd exposed the fungus to X-rays, and the X-rays could only damage the DNA. The X-rays would have no effect on the proteins themselves in the form of the enzymes. So they proposed the reason why these faulty enzymes existed was not because the enzymes themselves had become faulty, but because the X-rays had damaged the DNA, created mutations that led to a faulty enzyme. And this is how they made the connection between a gene and an enzyme. By mutating a gene, they created a mutated enzyme. And only a single enzyme was ever affected. So it's in the case of these mutations. And so they proposed that the product of a single gene was a single enzyme. And their proposal became a hypothesis, which became known as the one gene, one enzyme hypothesis. Now, very quickly, it became known that enzymes were proteins. This was something that was already reasonably well understood, but it was over a short period of time around this time frame that it was confirmed that enzymes were in fact proteins. So if genes were directing the synthesis of enzymes, and enzymes are in fact proteins, then it stands to reason that what genes are really doing is directing the synthesis of proteins. And so the hypothesis So the hypothesis then becomes one gene, one protein. But remember, at this point in time, people are also of the opinion, or at least a large number of people are of the opinion, that proteins are potentially the molecules of inheritance. So a lot of work is being done on proteins. And as people continued to research proteins, it became obvious that proteins were not made up of a single strand of amino acids. And many proteins, in fact, were not made up of single strands of amino acids. Many proteins turned out to be made up of many strands of amino acids. Like hemoglobin, it's made up of 4 strands of amino acids. And hemoglobin was actually a protein that was subject to a lot of research at this point in time. And so it became clear that as people investigated proteins and came to understand more about the structure of proteins, that we had to revisit what was considered to be a protein. And we revisited that when we talk about proteins back in chapter 5, when we stated that all proteins are polypeptides, but not all polypeptides are proteins, indicating that many proteins are actually made up of multiple polypeptides, each polypeptide being a strand of amino acids. And so once this is understood, once it's understood that proteins most frequently are made up of multiple polypeptides, you can't just say that one gene gives rise to a protein anymore. Because if a protein's made up of multiple polypeptides, a gene can't code for multiple polypeptides to give rise to a single protein. It must be that a gene gives rise to a polypeptide. So the final hypothesis states one gene, one polypeptide. And for our purposes at the moment, this is where the hypothesis currently sits. As far as most people are concerned, if we are going to state this hypothesis, we're going to state this hypothesis as one gene, one polypeptide. But as you'll come to see as we work our way through this chapter, we can make a really good case for rewriting this hypothesis all over again. And there are people thinking through how we might have to rewrite this hypothesis. But for the purposes of our class and for most people's purposes in most molecular biology books, if a hypothesis is stated creating the connection between genes and gene products, that hypothesis says one gene, one polypeptide. But that is not necessarily the case, as you'll see by the time we finish this chapter. But we'll just let that sit there right now. One gene gives rise to one polypeptide because it's a good way for us to start thinking about transcription and translation and the flow of information. So this is basically just pointing out the revision, but I've stated that for you. So once we talk about transcription and translation, we think in terms of the flow of information. We know that transcription is all about the production of an RNA molecule from DNA, and we refer to that RNA molecule ultimately as the messenger RNA because it contains the message, the information that's stored in the DNA molecule in a form that can then be recognized by and utilized by the ribosomes, the machinery that assemble the polypeptides, to actually go ahead and assemble the polypeptides. So the ribosomes read the information in the messenger RNA, and translate that into a polypeptide. And that leads us to this, again, flow of information. DNA to RNA in the form of the messenger RNA to a polypeptide. And the two processes are transcription and translation. Transcription, remember again, changing from one form of the language to a second form of the language. So we're going from one nucleotide form to a second nucleotide form. And then translation, change of language from nucleotides to amino acids when we go to polypeptides. And this is the flow in bacteria, DNA to RNA to polypeptide. And it is a direct flow. When we look at the flow in eukaryotes, it's a little bit more complicated. Number one, it can't occur in a single compartment as it would in a bacterial cell, because now the DNA is locked away inside the nucleus. So transcription takes place in the nucleus, but we know the ribosomes are out in the cytoplasm. That's chapter 6 if you've forgotten. So you might want to go back and take a look at that if you've forgotten the location of all of these organelles. So we know that the ribosomes are out in the cytoplasm, but the DNA is inside the nucleus. So transcription of necessity takes place inside the nucleus, but translation of necessity must take place out in the cytoplasm. So it's in two different locations. So that adds a level of complexity. Then as you'll see, while the product of transcription in bacteria is in fact the intact and ready to use messenger RNA, the same is not true in eukaryotes. The transcript is not a ready to go messenger RNA. The transcript, or what is referred to here in the diagram as pre-messenger RNA, needs to be processed. And there are a number of different steps involved in that processing, and we'll talk in detail about what that processing consists of. But in the end, again, still inside the nucleus, the pre-messenger RNA, the transcript, is processed to give eventually the intact, complete, and ready to go messenger RNA, which is then exported out of the nucleus so that it can be translated by the ribosomes. Is the flow still DNA to RNA to protein? Yeah, it still is. But there's a couple of steps involved in terms of the editing steps or the processing steps that are going to take place involving the RNA before it represents a messenger RNA that can be translated by the ribosomes. So there's a little bit more complexity. But nonetheless, the flow still is DNA to RNA to protein. So here it is. This is the, just make sure the pen's working. This is the dogma of molecular biology. And remember, this was proposed by Francis Crick. Of course, he proposed this after he and Watson and Wilkins received the Nobel Prize. So as a Nobel Prize laureate, people tended to take fairly seriously what he said. And so when Crick said the flow of information, this so-called dogma of molecular biology is DNA to RNA to protein, people were pretty willing to accept that. And of course, we know that this is pretty much the flow of information. Of course, we do know that it is possible to go back from RNA to DNA. So we just put that in. This is an additional piece of the puzzle that we now recognize. What is ironic here is that the ability to go backwards, the reverse transcription, and the reverse transcriptase, the enzyme that does the reverse transcription, is actually part of the virus. It actually has the toolkit that allows it to make the DNA copy of itself. And what makes the reverse transcriptase, the enzyme? The cell that the virus is infected. The RNA, that is the genome of the virus, includes the quote unquote messenger RNA sequence to make the reverse transcriptase. Ribosomes recognize the sequence and assemble the reverse transcriptase by reading the RNA sequence that's come from the virus. That then provides the reverse transcriptase enzyme that then makes the DNA copy that then is replicated by the cell's own machinery to make the double-stranded DNA that inserts into the chromosomes. So the virus is actually using the cell's own machinery against it, taking advantage of the fact that the cell recognizes the RNA sequence as a messenger RNA, makes the enzyme that then allows the initial DNA, single-stranded DNA copy of the RNA to be made by reverse transcription, which then that gets copied by DNA polymerase from the cell. So now the cell machinery is simply obliging the virus by turning that DNA into double-stranded DNA, which then gets inserted into the chromosome. So the virus really is using the cell's machinery against it. This is the process of reverse transcription. But again, remember, the primary flow of information and the predominant flow of information in cells is always DNA to RNA and to protein as proposed by Crick. So once this is recognized, once it is recognized that we have these two languages and that the flow of information is through these two languages, we then have to consider the quote alphabets of the two languages. So the alphabet of nucleotides consists of 4 letters, the 4 nucleotides. Whether we're talking about RNA or whether we're talking about DNA, it's still 4 nucleotides. When we think in terms of amino acids, however, the language consists of 20 letters, the 20 common amino acids that are found in all polypeptides. So when it comes to transcription, that's not a problem because we're just changing the form of the language. We're using the same alphabet, the nucleotides. And yes, I know that DNA has the T nucleotide and RNA has the U nucleotide, but the substitution is perfectly legitimate. And if you look at the 2 nucleotides, the T nitrogenous base and the U nitrogenous base are almost identical. So in terms of thinking in terms of nucleotides, yeah, we're thinking about the same language basically. But when we go from, when we do the translation from 4 nucleotide or 4-letter alphabet to a 20-letter alphabet, the first thing that you recognize is it can't be a direct letter for letter translation. Otherwise, there'd only be 4 amino acids in a polypeptide, and we know that's not the case. So if that is the case, if it's not a direct letter for letter translation, then obviously the solution has to be to create combinations of letters. Well, the smallest combination of letters is a 2-letter word. So how many 2-letter combinations can we make from 4 nucleotides? And the answer is 16. There are 16 possible combinations. That's a lot closer, but it's still not enough. So if it can't be a letter for letter translation and it can't be a 2-letter to each amino acid translation, then the smallest feasible word that would work has to be a 3-letter combination, what is known as a triplet, or to use the correct molecular biology terminology, a codon, a 3-letter nucleotide combination. So how many 3-letter nucleotide combinations can you make? A total of 64. And when that was recognized, which was very, very quickly, the effort then became trying to figure out if, number one, all the 64-letter combinations were used, and number two, if they were, what did they all stand for? Now, some of the combinations were relatively easy to work out. And in fact, the very first one to be worked out was UUU, which happens to be a codon for the amino acid phenylalanine. Why was that easy to figure out? Because all they needed to do was to make a polynucleotide of U's, a poly-U messenger RNA, and then provide all the necessary machinery, all the amino acids and everything that was required for translation, basically an extract from E. coli that contained everything that was necessary to do translation, and then provide the poly-U messenger RNA and see what was produced. And what they got was a polypeptide that contained just phenylalanine, which meant UUU must mean phenylalanine. The other combinations like that were relatively easy to figure out as well. So poly A, poly G, poly C were all fairly easy to figure out. But then once you started combining things together in all the other possible triplet combinations, it became a whole lot more difficult to figure out. And this was before the era of desktop computers. If you wanted to use a computer occupied a very large room and you had to book time for it. And you only found them at major universities and major research institutes and government departments. These were not things that people had ready access to. And so it required a lot of work and some very, very clever mathematics to figure out what each of the 3-letter combinations stood for. But in the end, after about 12 years' work, the full genetic code was figured out. So we now know that in the flow from DNA to RNA to protein, the DNA is red. And as we have seen previously when we talked about DNA replication, the template strand in the DNA molecule, or the template strand that represents the actual genetic sequence, is the 3' to 5' strand. So we can put here, as always, the DNA is red, 3' to 5', so that the template for transcription is the 3' to 5' strand. So if we're looking at this picture, this is our template strand. So this is the actual gene sequence that we're looking at. So when transcription takes place, you're going to make a complementary strand. So I'll put here the transcript is complementary to the template, with U replacing T, as we know it should. So here is the template strand. So A is complemented by U, C is complemented by G, and so on down. And that gives us then the messenger RNA. And notice the messenger RNA runs 5' to 3' because it is complementary to the template strand, and the template strand is red, 3' to 5'. So here is a little bit of a similarity to DNA replication. Remember, I indicated in DNA replication that the DNA polymerase always reads the template strand there, 3' to 5', and builds the complementary strand 5' to 3' by adding nucleotides to the 3' end. In DNA replication, however, the copying involves both strands, the leading strand and the lagging strand. Here, only one strand serves as a template. The other strand is often referred to as the non-coding strand. So the strand that runs 5' to 3' in this gene sequence would represent the non-coding strand. However, notice that we could read this strand 3' to 5' going in the other direction. And in some circumstances, particularly in bacteria, we know that both strands can serve as gene sequences depending on which direction they're read in. And the reason why this is the case is because in bacteria, you've got a very small DNA molecule. You've got a lot of genes to pack in there. So in many cases, gene sequences overlap going in both directions. So one gene would read 3' to 5' in this direction. A second overlapping gene would read 3' to 5' going in the other direction. So for one gene, this is the template, this is the non-coding strand. For the other gene, this would be the template, and this would be the non-coding strand. That is certainly possible, and we know that that sort of circumstance does exist within bacteria. Not so much in eukaryotes, however. Then once we want to translate the messenger RNA, we simply read them off in groups of 3, the triplets or the codons. So here are the triplets in this messenger RNA, and you'll notice that each one has a particular meaning. So UGG represents the amino acid tryptophan, UUU, first one ever figured out, means phenylalanine. GGC is glycine, UUUCA is serine. And so once you know the genetic code, and people who work in molecular biology very quickly learn the codons because they're using them all the time, and they recognize what each one represents. I still remember a little bit, although it's been a long time since I've had to do on-the-fly translations. So I'm a little bit sketchy on most of them, but I do know that UUU and a couple of the others I know still from memory as well, because they're really common. So now that we understand what the flow of information is, let's take a look at the genetic code. So here's the genetic code, all 64 possible combinations. And there are several things that stand out in the genetic code. Number one, we do need to remember that what we're dealing with is at best, collection of letters. I mean, that would be the analogy. Because remember, what we're dealing with is a set of nucleotides, and we represent each nucleotide by a letter. So that's why we write them out as letters. It's another reason why we use language terminology. And it helps us make sense of the way the genetic code works. So when we start thinking in terms of language again, how do we recognize where information is located in a collection of letters written on a page? Well, you know from English Comp that you can always identify the beginning of a message, beginning of a sentence, by looking for a capital letter. So once you identify a capital letter, you recognize that that's the beginning of the sentence, and then you use the groupings of letters to recognize words. And I'll show you later in the presentation, when we talk about mutations, just exactly how that works, turning a sequence of letters into words and recognizing them as words, because we can use them as an analogy for what goes on in mutations. So we need to be able to recognize too, in a sequence of nucleotides, where the information begins, what represents the beginning of the gene. So there is a single codon, and you can see it represented here, the AUG codon, which is referred to as the start codon. This is recognized as the beginning of the genetic sequence. This essentially is the flag that says this is where the gene begins. And it's really important that that be recognized as the beginning of the gene sequence. And as you'll see in the messenger RNA, there are ways to flag where the AUG is located for the ribosome so that they can identify where the AUG that represents the start of the message is actually located. Because once this is identified as the start of the message, this immediately sets in place how the remaining nucleotides are going to be read as codons, as groups of 3. We say that it establishes the reading frame, how you read the groups of 3 nucleotides as codons, how you frame them up into groups of 3 to represent codons. So the identification of the start codon, the recognition of the position of the start codon, and the fact that the start codon is a single codon with no confusion is really important for establishing the correct readout of the codons in the messenger RNA. Then just as it's important to recognize where the message begins, it's equally important to recognize where the message ends. So in language, we have 3 ways of stopping a sentence: a period, an exclamation mark, and a question mark. Now, don't read anything into this. There's nothing conspiratorial or anything else in this, but it just so happens that in the genetic code, there are also 3 ways of stopping the genetic message, what are referred to as 3 stop codons. And you can see them here. So here they are. These are the 3 stop codons. And these indicate to the ribosomes to cease translation when the ribosomes recognize these stop codons, to indicate that this is where the polypeptide ends, this is where the assembly ends, and they should stop adding amino acids. They are stop codons. And again, they have only one meaning, stop. So there's only one start codon, and it has only one meaning, start. Well, that's not entirely true, because it is also the only codon for the amino acid methionine, which means that the vast majority of polypeptides have as their first amino acid the amino acid methionine, unless there is some editing of the amino acids after the polypeptide has been created. We can talk a little bit more about that a little bit later. There is one other amino acid that has a single codon. Let me change color here. And you can see it here, tryptophan. Yeah, this is the amino acid that people talk about as being the amino acid in turkey that makes you sleepy after you've eaten Thanksgiving dinner. Whether that's the case or not, there's a lot of conflicting information about that. I strongly suspect that it's more the amount of food that you've eaten rather than the presence of the tryptophan in the turkey, but you can draw your own conclusion. But methionine and tryptophan are unique amongst the amino acids in that they have a single codon that codes for them. All the other amino acids have at least 2. Many have 4. 3 have 6. Change color again. So leucine has these 2 codons plus these 4. Serine has these 2 plus these 4. And then where do we have it? I gotta remember. Yeah, arginine has these 2 plus these 4. That tells you something about these amino acids. These are critical amino acids for the function of the polypeptides in which they are found, and particularly in the position within the polypeptide that they're found. So one of the ways that you can guarantee that that particular amino acid is in that particular position is to have multiple codons. And particularly when we start thinking in terms, let me change color again, when we start thinking in terms of the 4 codons that are grouped together like this, the only difference between these 4 is the letter at the end. So long as CGX is the codon, you're going to get arginine positioned in the polypeptide. Same with leucine. As long as you get CU, doesn't matter what the third one is, could be any one of these 4, you're going to get leucine in there. And the same with the serine up here. If you get UC in the messenger RNA, doesn't matter what the third one is, you're going to get serine put in. This has become recognized as what is referred to as the wobble base. And you can see that this is the case for many of the amino acids. So proline has these 4, and the only difference is the wobble base. Threonine has these 4. And again, it's the wobble base that is the only difference. Valine, the wobble base is the difference. Alanine, the wobble base is the difference. Glycine, the wobble base is the difference. Even where there are only 2 codons that code for the amino acid, it's still the the third nucleotide that is the difference between the 2 codons. So the wobble base idea is widespread in the genetic code. And it means that when we look at the code, there is a built-in redundancy. So we say that the code is redundant. Multiple codons for each amino acid. But it's not ambiguous. So when you look at a codon like this one, it only codes for threonine. There's no second definition for that codon. It only has one definition, threonine. That's what I mean by no ambiguity. Each codon specifies one particular amino acid. It doesn't specify multiple amino acids. So there is no confusion about what amino acid is meant by this codon. It means threonine. This codon means glycine, and so on. Each codon is very specific. So while there's redundancy, multiple codons for each of the amino acids, there's no ambiguity. Each codon only specifies a single amino acid. And that is very particularly true for the stop codons and for the start codon. Start codon means start. It doesn't mean anything else. Well, it does code for the amino acid methionine, but even there, it's still only coding for methionine. So while it does kind of do double duty, it really means only start, and it really means only methionine. The stop codons, they really mean stop and nothing else. So there's built-in redundancy, but there's no ambiguity in the code. And that's really important if we're going to accurately translate the messenger RNA into polypeptides. We also know that the code is universal. There are a couple of minor exceptions, but in a general sense, the genetic code is universal, which means when you transfer a gene from one organism to another, create a transgenic organism through genetic engineering, the organism in which you insert the foreign gene recognizes that gene and goes ahead and makes the appropriate protein because the code is universal. And so here's a tobacco plant that contains the enzyme luciferase, which is the enzyme that is used by the firefly to generate the light that it produces in the cells in the tail. And so because every cell in this tobacco plant contains the luciferase, it actually glows in the dark, just like a firefly. This pig contains a protein called green fluorescent protein, otherwise known as GFP. It comes from a deep water jellyfish, and it's used as a marker gene by genetic engineers to indicate that the piece of DNA that contains the genes that they're interested in has actually been inserted, because the presence of the green glow indicates that the gene for GFP has gone in, and so by extension, the whole segment of DNA has been inserted. And that's why this pig has a glowing nose and glowing trotters. And if you look really carefully, its eyes have a green glow. And if you could see the whole pig in the dark, you would see that it glowed green as well. Now, we do have two other proteins from deep water jellyfish called yellow fluorescent protein and red fluorescent protein that are used for the same thing as marker genes. But the only reason why they can work is because the universal genetic code. The fact that any organism can recognize a gene from another organism. In fact, those of you who are planning to go on and take our microbiology course will at some point in that course do an experiment where you will insert the GFP gene into bacteria and produce bacterial colonies that glow green, indicating that you've successfully inserted the GFP gene. It's one of the experiments that we actually do in our micro course. So if you're planning to go into the health sciences and you're taking our microbiology course in a future semester, you'll actually work with the GFP gene, this jellyfish gene, and insert that into bacterial cells, and you'll be creating bacteria that glow in the dark. It's kind of cool to see it, in fact. So we've been going for a while. Let's take a quick break. And this will be a full break, so I'll stop the recording after I give you the concept check questions and come back on the other side of the break with the answers. So Yarrad stated that humans inherit two characters. We would refer to those now as alleles. The term allele hadn't been developed at that point. For alkaptonuria, remember that's the condition that he was studying. And each parent must contribute a faulty version for a child to have the condition. This was part of what he observed. So if the parent had the condition, they could pass it on to their children. But if neither of the parents had the condition, but the condition was in the family, there was the potential for the parents to have children. And he's just applying Mendel's principle. And he indicated that the parents had to each provide a faulty version. So what does that tell us about alkaptonuria? Is this a dominant trait or is this a recessive trait? And what would be the polypeptide product of a 30-nucleotide poly-G mRNA? So this is an easy one. I'll see you on the other side of the break with the answers.